If you are interested in healthcare, you've probably heard about clinical trials, and you might also know these are studies designed to characterize safety and efficacy of a new medicine. This involves a series of studies for answering specific questions. For example, is the drug toxic to humans? What is the best dosage to cure disease without undermining patients' safety? Is it more effective than existing treatments? Can it be given alongside other drugs that are used for treating the same disease? To answer each question, clinical trials need to be designed properly, and the intention is to let the accumulated data speak, rather than subjective opinions of the doctors or the patients to override the decision. This can be influenced by many factors which could bias results. Rigorous clinical trials are set up to make sure this doesn't happen. At a very early stage, the problem of investigators and the patient's central interest is about safety. A dose given to a patient mustn't cause any severe side effects. This can be challenging in a phase 1 first-in-man study when very sparse data are available to inform how safe a particular dose may be. However, a phase 1 first-in-man trial is always preceded by preclinical studies that involve at least 2 animal species. Only once a clear understanding of safety and effectiveness in animals is established will it be appropriate to evaluate the drug's potential in humans. In current practice, animal data are used only to determine a safe dose to start with in a first-in-man trial. Once the human trial begins, animal data will be completely discarded. A very interesting question arising here is whether and how we can incorporate animal data into a phase 1 first-in-man trial for more efficient and ethical decision-making. A major objective of phase 1 clinical trials is to estimate the maximum tolerated dose in humans. They are commonly designed in an adaptive way. Patients entering the trial sequentially will be allocated with a dose that is estimated to be safe using all cumulated data. A statistical model is established to study the relationship between dose and toxicity. Had we already known how this dose toxicity curve is shaped, increased proportion of patients to be treated with the target dose can be expected. Unfortunately, this is not an easy task. Animal data must be translated first onto an equivalent human dosing scale properly, and due to intrinsic physiological differences, animal data can be inconsistent with human toxicity. Here, we have summarized the 4 types of plausible inconsistency of toxicity between animals and humans. As we can see, animal data may overpredict or underpredict the toxicity of the drug to humans. Any approaches that fail to discount inconsistent animal data quickly may result in ethical hazards that patients could be treated with overly toxic doses. IDEAS Research Project number 3 particularly looks at this issue, and we have proposed several Bayesian methods that are responsive to conflict between preclinical and clinical data. To find out more, please check our website and also see other interesting projects. This is Haiyan from IDEAS. Thank you very much for paying attention.